Hello, students, and welcome to my screencast on genetics. I really hope that you found this topic in the last couple of days of class to be fascinating. I think why do we look the way we do? Why do we have the traits that we do? It's just an awesome puzzle to figure out. It's so cool. So, real quick, just like all my other videos, this is free for students to help students. Nobody can profit from this, and neither can I. I use a lot of the Khan Academy content from it. I'm really digging Khan Academy, and the link is up there, which is also free. All right, here's a camel from Masira Island from our DO trip, and that camel's got a lot of genetics in it. And here's a picture one of our Courage buddies drew of one of our students showing a lot of genetics there. Let's dive right in. Okay, can you describe Mendel's experiments and what he figured out? I'm not going to ask you to just write a definition of the principle of segregation or the principle of independent assortment, but I would like you to know what Mendel did. Hopefully, you know, I forgot to animate this, that Mendel crossed various pea plants with different characteristics. This would be the P generation or the pure breeding generation. Let me see if I can get a pointer. So this would be the P generation. So these ones are always going to come out short, and these ones are always going to come out tall. So now that you know genetics, you would know that these are going to be homozygous, dominant, homozygous, recessive. Now you don't know which one's dominant or recessive. Most students will think it's the tall one, but we don't know if that's going to be the dominant allele yet. Then we get the F1 generation, and now you can see that the tall one is the dominant allele. Every plant comes out tall. Okay. Well, what's interesting is when you cross the F1 generation with itself, you then got 3 talls and 1 short. And here you got that classic Mendelian ratio. He didn't actually get 3-to-1, right? He did a lot of plants. He got 787 to 277. So he conjectured or figured out that this must be the ratio. So in other words, this short one was hidden. It was hidden within this tall plant, but it came out later on, just at a lower frequency. So let's see what else is going on here. If we predicted everything to be a blend, which a lot of students, when they first come to me about genetics, thinks everything are a blend, then everything would have come out medium, and they stay medium forever. Maybe they'd breed with something slightly taller and they come out a little bit taller, but that would be a blend. We didn't see that. Mendel didn't see that, right? He saw tall, short, all tall, and then 3 tall and 1 short. So he disproved the blending model, and we can disprove it as well. This is hard for us as humans to get our mind around because we often sometimes look like a blend of our parents. But in reality, we're thousands and thousands of Punnett squares going on in our body as we were made, and then we have complex genetic factors at play, like polygenic inheritance, that give us many genes contributing to a trait. So how did he do it? He made sure that the pea plants couldn't fertilize themselves, but that he could cross-pollinate them. Here's another example of the pea generation. So this would be color. This would be purple or violet flowers and white flowers. So they were always pure breeding or true breeding. So they always came out purple. They always came out white. When he crossed them, they all came out purple. So that tells you that the purple is the dominant allele. And then when he crossed them again, he got 705 purple and he got 224 white. P generation, F1 generation, F2 generation. What he's showing us is that traits can skip a generation and show back up. Here were some of the traits he looked at. We discussed them in class. Let's see what's going on, right? This is the important part. So we have, if we're going to cross yellow and green seeds, we don't know which one's dominant yet, but we cross them and we always get yellow. So then we're starting to think, okay, yellow is going to be the dominant color here. So we crossed those F1s again, and we got 3-to-1 with the green ratios. So that green is recessive. We can think of that in terms of alleles. This parent plant and this parent plant each give 1 allele, As we remember from our meiosis unit, you can only have one copy of each gene or one allele of each gene. Allele is a form of a gene in a sex cell. So here is its sex cell. Here's this sex cell. You put the two sex cells together and you get the diploid organism. Here they have a big Y and a little y. The big Y is the dominant gene. Don't think of it as, or the dominant allele, don't think of it as like this big Y overrides the little y. In reality, the big Y is producing enough protein for this phenotypic trait or for what you see to come out. Now, the F1 generation is going to produce its sex cells. It's going to produce big Y and a little y. Here comes the big Y and a little y. So this would be the top. This would be, you can say, the sperm or the egg. Remember, we're crossing two of these F1s, so big Y and a little y here. Then if you need a refresher on Punnett squares, you go over and down. So big Y, over and down, big Y, little y, over and down. We usually put the big letter first because it's just easier to understand that way, or easier for our human eyes. So big Y, and little y, little y. So if they were to ask you genotypic and phenotypic ratios, I just remember genotype stands for the letters, the gene. So we would have a 1 to 2 to 1 ratio. I didn't do that in class, so I definitely won't do that on your quiz too much. But 1 for big Y, 1 or 2 for big Y for the heterozygous condition, and 1 for little y. Notice that the phenotypic ratio is different because the heterozygous looks like the homozygous dominant. So 3 to 1. All right, we could also make that percentages. So how can we figure out what the genotype of a P is? We know that the greens are recessive, but how can we figure out if the yellow bag of seeds we have, and we know that they're all the same, if they're heterozygous or homozygous dominant? What we can do is what's called a test cross. And so we can run it with a homozygous recessive. And if it all comes out yellow, then that means this had to be a big Y and this had to be a big Y to make all yellow seeds, right? Because the green one's always going to contribute a recessive smaller one. If it came out half and half, then we know that this had to be a big Y and this had to be a little y. And that's what led to these half green peas. Okay, that has been a lot of me talking. I should have put some more checks for understanding in there. So see if you can understand this. Can you do this problem? Cross a heterozygous purple plant with a heterozygous purple plant. Purple is the dominant allele. White is a recessive allele. What is the percent chance that the plant would be purple? What is the percent chance that it would be white? Okay, hopefully you paused the video and tried it and you got something like this. So here's purple. I would have used peas, but this is what I could find with that could be shared on the internet for free. So big B, little b, big B, little b. So purple, 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 white. Hopefully you got something like that. Okay, that was a review of 8th grade. So how about 2 traits at once? Yay! So here's where we're going to have our 16 box squares. Remember, draw a big box, 3 lines down, 3 lines across, and you're going to get 16. So here is our P generation. It's always going to be well, not always, but our example we use in class and what I might use on your quizest will be a round yellow seed. It's always going to come out round and yellow. And over here would be a green wrinkled seed, It always comes out green and wrinkled. When you breed these 2 plants together, they always come out yellow and round. We know that is the heterozygous condition. Think about this. The only sex cells that this plant could produce would be a big Y and a big R. Remember that sex cells have 1 copy of each gene or 1 allele. This one would be little y, little r. So when they came together, it had to be big Y, big R, little r. Now we're gonna breed these F1s, and this is where I brought out new material to you in class. I don't think any of you had, or not too many of you had done dihybrids before. So can you figure out the possible sex cells for big Y, big R, little r? Remember that the sex cells go on the top and sides of a Punnett square. Gametes represent sex cells. They can only have one copy of each allele or the form of the gene. So in this example, you can only have one big Y or little y, one big R or little r. Do you remember our math trick? Hopefully you do, and hopefully you remember that it's called FOIL. So I'm going to write that here. So, oh, I thought I was going to write it there. Let's see if I can do it. Well, in the sake of time, let's see. I clicked off. Oh my goodness. Okay, in the sake of time, you're gonna pretend I wrote the word foil here. So let's go first, big Y, big R. Outer, big Y, little r. They did it a little backwards from us. Inner, little y, big R. Last, little y, little r. So you might say, hey, Mr. Gibney, they did it in a different order than we did. It doesn't matter as long as you get all the potential gametes there. So that's how you can solve it. And then you just, for a dihybrid, you're going to go and bring the same genes together. All right, so let's bring Y's together and R's together. Otherwise, we can't really read it. So let's look at this box. Be little y, big R, little r. So we know that it is a green round seed. So Mendel did this, and he did it repeatedly with different traits, and he always got this 9 to 3 to 3 to 1 ratio. And that's important, right? That is what we'd expect if these genes segregated independently of each other. In other words, if they behaved autonomous or they weren't hooked up to each other. They could behave however they wanted to. And so, can you connect that to our previous units? If you can, hurrah for you. Here it is. It's meiosis. So here's a big R and a little r, big Y and a little y. Here the gametes came out. Big R, big Y, big R, big Y. Little r, little y, little r, little y. But here, due to independent assortment of the homologous pairs of chromosomes, if we switch this one down here, we switch the big Y and the little y. It's a little hard to see, excuse me. Look at the different gametes it can ultimately give you. It gave you little r, big Y, little r, big Y. And over here, it gave you a big R and a little y. So that's how we got all the different ones. It really connects back to meiosis. So what happens if we don't get Mendelian ratios? Ta-da! Red hair and freckles, right? They often go together. Then we probably have what's called a linked gene. Right? If you have between 20 to 30,000 genes in every cell, and you only have 23 pairs of chromosomes, then chromosomes are going to have thousands of genes on them, on each of them. Okay, here's a practice dihybrid we did in class. This is a challenging problem. See if you can do it. You got to read through. You got to think about the terminology we use, etc. You got to write your own alleles. All right, how'd you do? Make sure you check the answer in Google Classroom. I've worked it out with you there. Okay, I taught you some shortcuts in class. So here I taught you that what's the probability of rolling snake eyes in a game? Snake eyes are 2 1s in a row. So what I'm really asking is what is the probability of a 1 here and a 1 here? So I want them together. I want to use the word and. That gives you the hint that you're going to use the multiplication rule and you're going to multiply the two. So the real answer here would be the chance of rolling snake eyes would be 1 over 6 times 1 over 6 or 1 over 36. Not very high. I wouldn't gamble if I were you. So here, if I ask you, what's the probability of getting a big B, big C, of getting a heterozygote from this cross between a heterozygote and a heterozygote? Well, I can do the 16 box dihybrid and then count them all up, or I could just do these two monohybrids. And so here, I've got the two monohybrids. I've got the B's. I cross them. To get the big B that I would want, it's going to be a 2 out of 4 or a 1/2 chance. Here I cross the C's, the big C, big C, little c. I cross them and I got 2 out of 4 or a 1/2 chance. And then I multiply the two together and I get 1/4. Okay, what are some other exceptions to Mendel's rules? Let me try and perk up here a little bit. I've had a little tonsil issue this weekend. And so I'm trying to get better so I can be the best teacher I could be for you this week. Can you describe incomplete dominance? Hopefully you remember from class, I told you that when you turn in incomplete assignments, nobody wins. So here, it's going to be an intermediate phenotype between red and white. It's going to be pink. So what is really happening is we're not producing enough protein pigments to be red, but we're producing more than are what needed to be white. Here is the Punnett square for it. And if you remember, if we cross two pinks, we can actually get a red and a white out of it. So that helps to disprove the blending hypothesis of genetics. Can you describe codominance? Right, so I said in class, a couple, everybody should be heard. Here's an example of this Rowan cattle. So it's going to express both the white and red coat colors. And here's the Punnett square for it. So crossing a red and a white and making Rowans. Can you cross two Rowans and see what you get? Why don't you try it out? Okay, can you describe multiple alleles? So multiple alleles would mean that there's more than two options. So everything we dealt with so far has been has been just two options. Here's the classic example. You can be A, B, AB, or O blood type. Hopefully you know your blood type. If not, ask your parents to see if you can figure it out. So this would be more than just two options. So that's an example of multiple alleles. And here's one with rabbits and chinchillas. And so they can have different coat colors and different alleles and all kinds of stuff. So this is kind of cool. They have to really figure things out if they want to be a rabbit breeder. Know your blood type terminology. So I'm just going to put this up briefly. This is from Arizona University that dealt with some genetics problems that I've been using in class. So here, an A person can be IA, or they can be the heterozygote IAI, the little I. This big I stands for the marker on the red blood cell, and then A stands for that type. It's just what we use when we write genetics of blood. Okay, can you describe sex-linked genes? So for this one in class, I kept using the example of hemophilia. See if you can describe it. Okay, here we have, it's going to be a recessive disease. So if they get their recessive copy, they won't be able to clot their blood. They'll just keep bleeding all out if they get a cut. Or internally. So here's the father, he's unaffected. Mother is a carrier. Notice who is affected. None of her daughters can be affected, right? They have to get this big H from dad. So this daughter is a carrier but is unaffected, and this daughter is unaffected at all. This son is unaffected, but this son has hemophilia. And so this happened in the royal families in Europe and Russia, and it led to the rise of Rasputin. And so hopefully we'll get that in class. I thought we would have by now, but it's okay. We've been learning a lot of stuff. It was also shown by fruit flies, which I think I have on the next slide, which I do. So for fruit flies, we use slightly different terminology. The W stands for white. The plus stands for the wild type, or what you see in the wild. So here, if we cross a red-eyed female and a white-eyed male, we're going to get red-eyed female, red-eyed female, red-eyed male, red-eyed male. So these are all going to be red eyes. This is the F1 generation. Now let's cross that F1 together, the red-eyed female and the red-eyed male, and we're going to get red-eyed females. But look over here, we're going to end up getting some white-eyed males. So if this wasn't on the sex chromosome, then you would expect we would expect this to be on a white allele here, and so we would expect to get more of a mix between females and males. But because we don't, Thomas Hunt Morgan and his graduate student Alfred Sturdivant, I love to smoke pipes, figured out that it's a sex-linked trait. So I got some bonus research for you, a couple bonus questions to check out. Do you remember when we talked about calico cats or Siamese cats? How about some of these? These are pretty cool. If you take my AP class, we'll talk about these. Pleiotropy. And I gave you a couple examples to look up. Epistasis, lethal alleles, polygenic inheritance, and epigenetics. This one, I keep meaning to show you the video of the twins and talk about the astronauts, and hopefully we'll get to that soon. Okay, last but not least, which is good because I'm running out of steam with my tonsils here. We're looking at pedigrees. So we use Roman numerals. This would be I-1, I-2. This would be 2-1, 2-2, 2-3. It just helps us to label everything. What I told you in class, and this might have annoyed you, was to be one with the pedigree and try and figure out what's going on. If you try and memorize a bunch of rules, you can, and it'll get you by on a test, but it'll be tough to memorize those rules and remember them later in the year. So here's the 3 diseases I'm going to quiz you on: autosomal recessive, autosomal dominant, and X-linked recessive. I will not quiz you on X-linked dominant. Here's an autosomal recessive pedigree. The way we can tell is that it skips a generation. It skipped 8, 9, and then their child, 17, had it right here. I do apologize that this pedigree is labeled in numbers that just keep on going instead of the Roman numerals. Sometimes you'll see that. So could it be sex-linked? Well, let's look right here. This lady, she could get an X from dad that's affected by it and an X from mom that is not. So that could be big H, little h. This dad would then have to be affected by it. It had to be little h in order for this child to have it. So this cannot be sex linked. So the way to look for sex linkage is look at a female and then look at her father and look at her sons and her daughters, and you can figure it out. So usually autosomal recessive traits skip a generation and they affect males and females equally. Here's some examples of autosomal recessive diseases you can check out. And we're doing, our deep dive into sickle cell anemia. All right, autosomal dominant has to be found in every generation, otherwise it dies out. It usually goes to half the children regardless of sex. And so here we can tell that it's autosomal dominant because these two have the disease, but yet they have a child who doesn't. If they were recessive, little a, little a, little a, little a, then all their kids would have to have the disease. So that's one way to tell if it's autosomal dominant. Dwarfism is one, and Huntington's disease is another. And last but not least would be X-linked recessive pedigrees. So for us, this was hemophilia we talked a lot about, and color blindness. Males are going to be more often affected than females. And affected fathers can't pass on to their sons because they can only give their sons a Y. Okay, see if you can figure out the pedigree. Pause here. Well, if it gave it away to you, because there's kind of a bunch of X's in it, sorry about that. This would be color blindness. And so we can look at it, we can figure out that it's sex-linked recessive. If this lady has it, she got one X from dad that has it. So we could say XC, this would be an XC. And then her kids are going to have it, right? All of her males. One thing that's interesting to note, if we say something is sex linked, we can't rule out autosomal recessive 100%. So we'd have to do some genetic testing for that. Okay, how about this one? So here, check it out. This is pretty cool. This is twins, like my twins. Hopefully you got autosomal recessive, so it skips a generation. Let's see if it could be sex linked, right? So mom would be a carrier X, big H, X, little h. Dad would have to be affected by it, so this cannot be sex linked. Okay, how about this one? If you caught me, it is the same. I had trouble finding one with autosomal dominant. I probably should have just drawn one, sorry. But this is dominant because 5 and 6 have a kid that doesn't have the disease. So this could be Huntington's disease. And this one, check it out. This is an example of recessive. Let's see if it could be sex linked. So this female would be X big H, X little h. That would be X big H. That works down here. Dad gave her the little h. Mom would have to be a carrier. So this could work. Mom carried to that one. Gave it there. If this was a little h, you see me kind of thinking everything out. Here, dad gives the Y. Mom could be a carrier. So we can't rule out sex linked here, which is kind of interesting. So unless I'm missing something, and if I am, point it out to me in class, and I'll give you a cookie. But it's definitely recessive because it skips this generation right here and goes here. All right, students, that was a lot. I wasn't my usual peppy self, battling an illness right now, but we're going to get through it. And, I'm really hoping, I just wish there were more days to have class with you because I have so many activities and meaningful phenomena and next generation science gadgets to go into with genetics. We're just going to run short on time. Sorry about that. Okay. Have a great night for your test.